There is a wide diversity in living organisms in our biosphere. Now a question arises that are all living organisms made of same chemicals, that is elements and compounds? If we perform elemental analysis on a plant tissue, animal tissue or a microbial paste, we obtain a list of elements like carbon, hydrogen, oxygen, several others and their respective content per unit mass of a living tissue. If the same analysis is performed on a piece of earth crust as an example of non-living matter, we obtain a similar list. What are the differences between the two lists? In absolute terms, no such differences could be made out. All the elements present in a sample of earth crust are also present in a sample of living tissue. However, a closer examination reveals that the relative abundance of carbon and hydrogen with respect to other elements is higher in any living organism than in earth crust. My dear learner, a very warm welcome to all of you. Myself Ankita, and in this session we will discuss about the biomolecules. The molecules that supports life, and during the discussion we will talk about metabolites. All the primary metabolites and secondary metabolites, biomacromolecule and polysaccharides. Have a look at the comparison of elements present in the non-living matter and living matter. If we compare the elements that are present in non-living and living matter, here is a picture that depicts percentage weight of earth crust and human body. The hydrogen atom, which is 0.14% weight of earth crust, and in human body it is 0.5%. But whereas carbon, it is 0.03% weight in earth crust and in human body it is 18.5%. Whereas oxygen, it is 46.6% weight of earth crust and in human body it is 65%. Nitrogen, that is very little percentage weight in earth crust, but in human body this is 3.3%. Sulfur, this is 0.03% and in human body this is 0.3%. Sodium 2.8% and in human body it is 0.2%. As comparison to magnesium, this is only 2.1% weight in earth crust, whereas in human body it is 0.1%. Silicon, that is having percentage weight in earth crust is 27.7%, but in human body it is negligible. We can continue asking in the same way, what type of organic compound is found in living organism? How does one go about finding the answer? To get an answer, one has to perform a chemical analysis. We can take any living tissue, a vegetable or a piece of liver, etc., and grind it in trichloric acid using a mortar and a pestle. We obtain thick slurry. If we were to strain this through a cheesecloth or cotton cloth, we would obtain two fractions. One is called the filtrate, or more technically you can use the word the acid soluble pool, and the second the retentate, or you can say the acid insoluble fraction. Scientists have found thousands of organic compounds in this acid soluble pool. All the carbon compounds that we get from living tissues can be called biomolecules. The molecules that are involved in the maintenance and metabolic processes of living organisms. However, living organisms have also got inorganic elements and compounds in them. How do we know this? A slightly different experiment has to be done. One weighs a small amount of living tissue, say leaf or liver, this is called wet weight, and dry it, then all the water evaporates. The remaining material gives dry weight. Now if the tissue is fully burnt, all the carbon compounds are oxidized to gaseous form, that is CO2 and water vapor, and are removed. What is remaining? The remaining product is called ash. This ash contains inorganic elements like calcium, magnesium, etc. Inorganic compounds like sulfate, phosphate, etc. are also seen in this acid soluble fraction. That means you can say that this acid soluble fraction contains most of the inorganic compounds, which include sulfate, phosphate. Therefore, elemental analysis gives elemental composition of living tissues in the form of hydrogen, oxygen, chlorine, carbon, etc. While analysis for compounds give us an idea of the kind of organic and inorganic constituents present. That means if we analyze the compounds, that will give us an idea whether this is a organic compound or an inorganic constituent which are present in the living tissues. If they are present in the living tissues, they can be sugars, amino acids, fats, oils or nucleotides. From a chemistry point of view, one can identify functional groups also like aldehydes, ketones, aromatic compounds, but from a biological point of view, we shall classify them into amino acids, nucleotide bases, fatty acids, etc. So, biomolecules are molecules that occur naturally in living organisms. Biomolecules include macromolecules like proteins, carbohydrates, lipids and nucleic acids. It also includes small macromolecules like primary and secondary metabolites and natural products also. So, you can say that a biomolecule consists mainly of carbon and hydrogen with nitrogen, oxygen, sulfur and phosphorus also. Biomolecules are very large molecules of many atoms that are covalently bound together. There are 4 major classes of biomolecules. You can say first is carbohydrate, lipids, proteins, nucleic acids. Now, what are the primary and secondary metabolites? The metabolites are the products of metabolism. Now, what is metabolism? Metabolism is a biochemical process which is an important process for the energy production in the body. The term metabolite is also usually restricted to small molecules. Metabolites have various functions including fuel structure, signaling, stimulatory and inhibitory effects on enzymes, catalytic activity of their own, usually as a cofactor to an enzyme, in defense and interaction with other organisms. For example, pigments, odorants and pheromones. The metabolites may be primary metabolites or secondary metabolites. A primary metabolite is directly involved in normal growth, development and reproduction. Ethylene is an example of a primary metabolite produced in large scale by industrial microbiology. A secondary metabolite is not directly involved in those processes but usually has an important ecological function. For example, they act as antibiotics and pigments such as resins, terpenes, etc. Some antibiotics use primary metabolites as precursors such as antinomycin, which is created from the primary metabolite tryptophan. Some sugars that are metabolites, examples include fructose, glucose and these are in the metabolic pathways also. In animal tissues, one notices the presence of all these categories of compounds. The primary metabolites are involved in the growth and development of the body. However, when one analyzes plant, fungal and microbial cells, one would see thousands of these compounds other than these compounds which are called primary metabolites, but we have other compounds other than these primary metabolites, they are called secondary metabolites. For example, alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums, spices, etc. Here is a table which is depicting some of the secondary metabolites. Pigments include carotenoids, anthocyanins, etc. Alkaloids include morphine, codeine. Terpenoids, they include monoterpenes, diaterpenes. Essential oils include lemongrass oil. Toxins include Altrine, ricin, etc. Lactins include concavalin A. Drugs include vinblastine, curcumin, etc. Some of the polymeric substances are also secondary metabolites. While primary metabolites have identifiable functions and they play known roles in normal physiological processes, we do not at the moment understand the role or functions of all these secondary metabolites in host organisms. However, many of them are useful to human welfare. For example, rubber, drugs, spices, sands, pigments. The differences between primary and secondary metabolites are basically the primary metabolites formed as a result of energy metabolism, and they play important role in growth of an organism. These are widely used in industries for cosmetic production, food production, alcohol, beer, wine production. But secondary metabolites are modified primary metabolites. They do not play a role in growth and development. The secondary metabolites act as antibiotics. Erythromycin, bacitracin, these are important and are the common antimicrobials. Pigments like carotenoids, anthocyanins, alkaloids, morphine, codeine, terpenoids, essential oils, toxins, lactins, drug, polymeric substances, rubber, gums, cellulose, these all are the examples of secondary metabolites, but other than primary metabolites. The primary metabolites are essential to cell growth. They are involved directly in metabolic reactions such as respiration and photosynthesis. Most of the primary metabolites are identical among most organisms, whereas secondary metabolites are numerous, widespread and unlike the primary metabolites. The secondary metabolites are derived by pathways in which the primary metabolites involve. That means you can say the secondary metabolites are the products of the primary metabolites. Therefore, secondary metabolites are considered as the end products of the primary metabolites. The primary metabolites are produced during the growth phase of cell, while secondary metabolites are produced during the non-growth phase of the cell. The secondary metabolites are accumulated by plant cells in very small quantities than the primary metabolites. The growth phase where primary metabolites are produced is sometimes called trophophase, whereas the phase during which secondary metabolites are made is called idiophase. Most of the secondary metabolites are involved in defense reaction. As I already have told you, they act as antibacterial, antimicrobial, etc. But unlike the primary metabolites, proteins, carbohydrate, lipids, these are the main primary metabolites, whereas secondary metabolites are alkaloids, phenolics, sterols, steroids, essential oils and lignins, etc. Now let us come to biomacromolecules. What is actually a biomacromolecule? A macromolecule is a very large molecule such as protein, commonly created by polymerization of smaller subunit. You can say the small subunits, they polymerize to form large molecule. This is called biomacromolecule. They are typically composed of thousands of atoms or maybe more. The most common macromolecule in biochemistry are biopolymers. These include nucleic acid, proteins, carbohydrates and polyphenols, and large non-polymeric molecules such as lipids. The synthetic macromolecules include common plastics and synthetic fibers as well as experimental materials such as carbon nanotubes. There is one feature common to all those compounds found in the acid soluble pool. As I already defined earlier that a compound is divided into acid insoluble pool and acid soluble pool. So, I am talking about the feature that is common to all those acid soluble pool. They have molecular weight ranging from 18 to 800 daltons. The acid insoluble fraction has only four type of organic compounds, that is proteins, nucleic acids, polysaccharides and lipids. These classes of compounds, with the exception of lipids, have molecular weights in the range of 10,000 Daltons and above. For this very reason, biomolecules, that is chemical compounds found in the living organisms, are of two types. One, those which have molecular weight less than 1000 Daltons and are usually referred to as micromolecules or simply biomolecules, while those which are found in the acid insoluble fractionaire with molecular weight above 10,000 Dalton, they are called biomacromolecules. So, basically two divisions, one which are having molecular weight less than 1000 or which are having above 1000. The molecules in the insoluble fraction, with the exception of lipids, these are the polymeric substances. Now, a question arises here, why do lipids whose molecular weight do not exceed 800 Dalton come under the acid insoluble fractionaire, that is macromolecular fraction? Actually, lipids are indeed small molecular weight compounds and are present not only as such but also arranged in structures like cell membrane, other membranes also. When we grind a tissue, we are disrupting the cell structure. A cell membrane and other membranes are broken into pieces and these form vesicles which are not water soluble. Therefore, these membrane fragments in the form of vesicles and these get separated along with the acid insoluble pool and hence in the macromolecular fraction. Lipids are not strictly macromolecules. The acid soluble pool represents roughly the cytoplasmic composition. The macromolecules from cytoplasm and organelles become the acid insoluble fraction. Together, these represent the entire chemical composition of living tissues or organisms. So, to summarize, if we represent the chemical composition of living tissue from abundance point of view and arrange them class wise, we observe that water is the most abundant chemical in the living organism. Here is a table which is showing you the average composition of cells. Water having percentage of total cellular mass, that is around 70 to 90%, whereas proteins, these are 10 to 15%, carbohydrates 3%, but in comparison to lipids are 2%, nucleic acid 5 to 7%, ions 1%. So, you can say that water is in large quantity. So, in nutshell, you can say that a compound is divided into two categories. One is acid soluble pool and other is acid insoluble. You can use another word for acid insoluble or you can say retentate. The acid soluble pool includes amino acids, monosaccharides, disaccharides, fatty acids, glycerol, nucleoside, nucleotide, nitrogenous bases. But in acid insoluble pool, there are only four categories: proteins, nucleic acids, polysaccharides and lipids. The acid soluble pool having molecular weight 18 to 800 Daltons, but in acid insoluble pool, there is molecular weight above or around 10,000 Daltons, and basically the acid soluble pool are referred to as micromolecules, but acid insoluble, they are known as macromolecules or biomacromolecules, and they are the part of cell membrane, cytoplasmic organelles, etc. The acid insoluble or retentate, they are again divided into two categories. They are polymeric and non-polymeric biomolecules. The polymeric biomolecules having weight more than 1000 Daltons and non-polymeric with molecular weight less than 800 Daltons. The polymeric retentate or acid insoluble pool includes proteins, nucleic acids and polysaccharides. The proteins which are having polymers of amino acids, they are linked by peptide bonds, and they include further the polymers of several other amino acids. The nucleic acids include DNA and RNA. What is DNA? DNA here is polymer of deoxyribonucleosides, and RNA, that is polymer of ribonucleotides. The RNA is linked by phosphodiester bond, DNA is also linked by phosphodiester bond. The non-polymeric biomolecules include lipids. Lipids are the esters of fatty acids with glycerol. They are divided into two categories. They are simple and compound. Simple include oil, fat, waxes. Compound includes phospholipids, glycolipids and sphingolipids. So, to summarize what we have discussed about biomacromolecules, metabolites, further primary metabolites, secondary metabolites, their function, their role, their actual structure and also the necessary products also. Thank you.